That's really very kind. I hope I can keep up with the expectation that Emma has. Yeah, and it's a pleasure to talk here because indeed, since maybe the beginning of the 2000s or so, we started to work with neonates. And since then, actually, now all members of the group actually working. There's nobody working anymore with adults. And in our case, actually, adult mice, we're very much doing mouse work. Sometimes we touch humans. So I think Emma introduced this. This is like kind of, if you wish, the interface that we're interested in, which is the gut surface. And the point maybe to remember again is that this is not a passive surface, but rather very active. So what looks stable is in fact very dynamic. If you would remove any of the interplaying factors, it would get out of balance, and then you would end up with some sort of disease. And the players here involved is the microbiome, which is maybe the focus today, but also the epithelium, which is complicated on its own and has many different types of cells, and the immune cells underneath. And they speak to each other, all the three, permanently. And so this is then physiology. And on top comes maybe infection, and again, microbes. And so we do work with microbiota, but we also have a focus on certain types of enteric pathogens, among them Salmonella and EPEC as gram-negative bacteria, Listeria, and as a parasite, Giardia and rotavirus as an example for a virus. And I think the next three following slides I shouldn't be presenting because honestly I didn't know this this foundation. So I wasn't aware of there's something that an institution that only works with infants. So I wouldn't have to present this, but because I normally have to defend that I work with neonates, right? People say, yeah, why do you do this? You should, everybody works with adult mice. Why would you touch neonate mice? And so I have the following three slides. And so step back, get one step back, and then you will understand why I'm doing this. Because so the first reason why we work with neonates, as is the reason for the microbiologist. And the reason here is that if you look into mortality from infection, and this is what you can see here, it's clearly, do you have any point? I would have to do it this way. Yeah, it's clearly the neonates actually die from infection, right? So overall, it's infant mortality that is driven by infectious diseases, and in particular, the first year of life, and even more so, the first 30 days of life. If babies die, they have a very high probability to die from infection. So infection research really makes sense in neonates. I would reply to people that work with adult mice. So you guys should work on neonates and rather, switch from adults to neonatal work. And the second question is maybe to put this a bit provocative, to point that the Janeway is an immunological textbook that explains many things, but it doesn't explain why you survive being born. So this is very provocative. But it basically, what I mean with this is that you transit from placental life, right, protected, and Emma referred to this, to postnatal life, which is heavily exposed. And Emma had some nice images on how this happens. And it is really hard to think how this can happen. How does the innate, how does the adaptive immune system cope with this? And how does it come that we all end up being, as we sit here, rather healthy? And the interplay between the three players and at the gut mucosa does work well. And I think we cannot explain this yet. So I think there are many open questions here that need to be answered. And again, conferring, to the microbiome, to the innate system, and the adaptive immune system. And maybe to counter dict a bit a certain type of misconception that I sometimes see is that neonates are not small adults. So don't think they're just smaller. Otherwise, they're the same. They're very, very different, which is good for us because this is the way we do science. And the other thing is that neonates are not immature. They're not deficient in something. They're not sick per se. They're optimized to be neonates. So, and Emma referred to this. So neonates have a very different environment, very different demands when it comes to energy, when it comes to food, when it comes to many things. So we have to consider neonates to be perfect, but to be perfect as a neonate. So yeah, okay. I think this is just to because I sometimes hear this and I think I don't want any more. And the third reason, again, goes beyond infancy. And it's very interesting. And that's it's a hypothesis. And it's the hypothesis of the window of opportunity. And basically what this means is it seems that certain factors prime the immune system to act in a certain way. And they do this in particular during early life. And this is called the window of opportunity. It's a time window where these factors can act on the immune system. And then put this in a certain position to then lifelong act in a certain way. And this is non-redundant. So if you miss these factors during infancy, you cannot cope with this later. And it's interesting hypothesis, because it would mean that factors in early life would influence disease susceptibility all lifelong. And this refers to mostly to immune-mediated diseases like allergies or asthma and some metabolic diseases. But again, I think very interesting to think this way when we consider neonates. And so this is kind of what we do in the group, right? So we work on the microbiome, working on the diet of the neonates, so mostly breast milk, which is very interesting. And then innate changes of the adaptive immune system, and structural changes that the epithelium kind of undergoes. And you have a little cartoon here, so you can see that this is actually quite different. This is a bit more potent in mice, almost stronger in mice, which have a much shorter gestational period. And this is the reason. So they have just 3 weeks in utero and then get born. Humans have 9 months in utero, right? So they're more mature when they're born, which means they're more different, or explains why there are more differences. Nevertheless, mice are mammals and they get born and survive this. So now, maybe to start with this, again, a provocative question: when actually does colonization start? I mean, Emma suggested birth, and I completely go with Emma, but there have been papers that suggest otherwise. So the so-called sterile womb hypothesis or paradigm has been established, and really interesting, in something like 100 years of work. So Escherich, this is the guy who gave Escherichia coli the name. Theodor Escherich was a guy in Munich. So there's a paper from 1885 until 1984. 100 years of research established that this, fetuses are sterile. And then came a couple of papers published in, very renowned journals that promoted the placenta is colonized and fetus is actually colonized. And so how come? How could 100 years of research miss this? And the problem here is, and that's kind of interesting, and I think conceptually maybe we can learn something, is probably a new technique, and it was the next generation sequencing activities. So basically what these papers reported was bacterial DNA in the placenta or in these fetuses or embryos. And we have to remember, DNA is not microbes, it's just DNA. And there was some more inconsistencies, but nevertheless, obviously the data was strong enough that these journals accepted these papers to be published and the idea to be promoted. I think this has been contradicted in more recent papers now, and I think kind of the common sense is now we would again, say the sterile womb hypothesis is true, and when we're born, we're sterile. Nevertheless, just to bring this up, because I think when you think about how science works, this is a very nice example. And sometimes it takes quite a time to get people, back to thinking. And the strongest argument, there are several arguments why obviously, we're sterile when we're born. Germ-free mice, and Emma has presented them. We can produce germ-free mice when we operate pregnant female animals that are colonized and then re-derive them in a sterile fashion. We actually can do this with humans. And this is the case. These are the bubble childs. So this child was born from a mother that had previously born a sibling that died from immunodeficiency. And so she delivered the baby, and this was the pregnant mother. And so the baby was delivered in a sterile cabinet and then transferred, and grew up the first days of life under completely sterile conditions, like a germ-free animal. And microbiological testing could prove that this child was actually pretty sterile. And obviously this would not work if you're colonized prior to birth, right? So this is maybe the strongest argument they can come up with. But if that's the case, so how does it then continue? So then the microbes come as, maybe, the baby sits on the floor and is drinking milk together with cats next to them. And so it's picking up microbes from the environment. But how would that work? And so we can look at different types of aspects of this microbiome and look at the kinetic. And it's actually quite interesting. So we could look into density, which would be number of microbes per volume. And here, and this is human data, it's a very old paper, but still very nice, I think. So I just look into, in feces, number of bacterial genome copies. And what they see is within 2-3 days in the feces per gram, the density reaches threshold, so it's up there. So quantity-wise, colonization is extremely rapid. Okay, think immunologically. This is surprising that you survived this, right? I think this is really striking, but this seems to be the case. Now, when we think about diversity, which means now different types of bacteria with different names, present in the gut, this is very different. So here it takes something like, and now we're counting years, not days. We are maybe at 2-3 years in humans to reach threshold of diversity. We can think about compositional succession of bacteria. This is what we know the least of. And this means that, we have different types of bacteria that bloom and then decrease and come and go. And there seems to be a certain logic behind it, but this is very chaotic otherwise as well. So kids are quite different as animals are, individual animals are. And I don't think we really understand, why some do grow and some others disappear or are reduced in abundance. And maybe the last one, the least exciting, is anatomical specialization. It means that if you're born, your flora looks, microbiome looks pretty much everywhere on your body the same. And after a while, this becomes more distinct. Now, we also looked into this. And again, in mice you can do things that you cannot do with humans. And it's kind of surprising. So at the upper part, you see density. And if you look into the bacterial genome copies in the small intestine and the colon over time, what you see in the colon, that's a bit what you expect. It rises and then it reaches a certain, maybe it peaks a bit, but then it reaches a certain threshold. But it looks very different in the small intestine. If anything, the small intestine is colonized extremely rapid. So this is day 1 after birth, 24 hours after birth. And if anything, there is no increase further. If anything, there's actually a decrease. So neonates tend to have more microbes in the small intestine than adult animals. And with culture is complicated in that sense here, but we would at least confirm that there's not less. Now, when we look at diversity, and this is metagenomics, so here we're not sequencing just amplicon, but just the whole genomes of anything that's there. And then we can very nicely also look at strain level and something. And here we have the outer ring, we have the adults, and the inner ring, the neonates. And again, we see something actually surprising, which is that all the highly abundant species in the adults, which are kind of the black, if you wish, the black bars here, they're also present in the neonates. It's rather the kind of low abundant species that are absent in neonates. So it's not completely different, but we do lack lots of strains, but it's kind of that the highly abundant ones are found. And this explains why the species richness, that's a bit similar to diversity, is the same, is different. So it's much higher in adults. And the BOTA score, which predicts, and that's just for the immunologists, kind of the number of immunodominant epitopes that are provided by the microbiome, again, is higher. But if you normalize this kind of to the abundance of a given taxon, then actually this becomes pretty much the same. So a given immune system at any spot would see pretty much, sufficient immunodominant peptides and proteins to react against it. And this is also what we see when we do proteomics from the gut content. This sounds a bit crazy, but we've been interested in, in neonates versus adults, how much protein load is there? How many different types of proteins would you find if you would be an immune system that wants to look into, seeing different types of epitopes? And if, there's a great overlap, this comes obviously from the highly abundant species, which are the same. And then we see differences, but, there's no significant difference. If anything, neonates, certainly don't have less, peptides. Again, proteomics is not very sensitive as a method, which I think explains what we see. But we would think, it's not just the absence of peptides or absence of immunodominant peptides that explains something that I will come to later. So then maybe the next question, if there's so much chaos after birth and things come and bloom and go and increase dramatically, you could ask the question, and it's so important for the host, how much does the host interfere with this? And we know, for example, breast milk is extremely strong in doing this. In humans in particular, we have human milk oligosaccharides, and they're really drivers of bifidobacteria. The strength is absolutely amazing. We can, in an adult individual, it's really hard to change the microbiome. It's really tough. But what you can do is you give these human adults human milk oligosaccharides, what is in breast milk, and then you drive actually bifidobacterium growth in a human adult gut. You can do this. With no other treatment, this is possible, otherwise maybe antibiotics. So this is how strong they are. So this is obviously the host tries very hard to influence the microbiome in humans. Now, mice don't have these. In mice, you don't have these oligosaccharides. And so maybe there are other factors. And we looked into one, and this came up when we did the expression screen, neonatal epithelium versus adult epithelium. We found 2 of the TLRs, toll-like receptors, receptors for certain PAMPs, so structures from microbes, were differently regulated. TLR3 was down in neonates. This recognizes double-stranded RNA, like from viruses. But TLR5, the receptor for flagellin from bacteria, was up in neonates. And when we kind of did the kinetic, that's very clear. So it's very much up in the epithelium, and it goes down. It doesn't disappear. And in this paper here, they showed that this is actually pilot cells that keep expressing it in the adult. Now, this age-dependent regulation is only in the epithelium. It's not seen in immune cells, which is interesting. So there seems to be a very particular aspect here. And then we thought it's something to do with infection. And so, try to do infection in neonates, whether this TLR5 expression would protect the neonate. And we couldn't really see this. And it took a while until we understood that maybe it's nothing to do with host defense to infection, but rather colonization regulation. And so we did a competition experiment, something that is an extremely sensitive tool. So we put into a mouse a 1-to-1 ratio of flagellated and non-flagellated salmonella. Just put together 1-to-1. So, one has the ligand for the TLR5, the other one doesn't. And then we look over time. And then we see over time that obviously the non-flagellated is favored. This is only seen in neonates, not in adults, which fits the kinetic of TLR5 expression. This is only seen in the presence of TLR5, and only if TLR5 is also expressed by the epithelium. That's important because immune cells can also make this. And now this suggests that indeed that TLR5 on the epithelium in a neonate would kind of counter-select flagellated bacteria in this situation. But Salmonella is not microbiome, clearly. So the next step would be we need to show that it does influence the microbiome. And so how did we do this? We took advantage of the fact that previously Andrew Gewertz had shown that TLR5 knockout adults have a different microbiome. And now we would, our hypothesis, of course, was that that's because enteric TLR5 couldn't shape the microbiome in a certain way. So this is the difference. So we have a microbiome from a TLR5 knockout mouse and a wild-type mouse that shows a certain difference. And now we put these 2 microbiomes in individuals that are either neonates or adults, and then look over time. And what we could see, maybe start with the adult that do not express TLR5, that the distance between the 2 different microbiomes that are different remained the same over time. Whereas in neonates, it became more similar. So this UniFrac, which kind of is a model to show distance, so similarity, which would confirm that the presence of epithelial TLR5 is able to make these 2 microbiomes that are different because of the lack of the signal more similar. Okay, so maybe yes, the host may try to shape the microbiome. And there's another effort where we tried to think more globally. What else could influence the microbiome composition in neonates? We thought about metabolites. And Emma had an example of metabolites before. So what we did is we took animals in which we did the microbiome analysis, and this is what's shown in this plot. So we have day 1, day 7, day 14, 21, and 28. You see the microbiome changes, and there's a major jump at weaning when they start to eat solid food. And in the identical mice, we took the liver and did metabolomics. And the interest was that which metabolites are age-dependently different. And the ones that kind of stood out here were like bile acids. Okay, we didn't know this, bile acids, so we start to read. I thought, okay, that's interesting. They're secreted by the liver into the gut lumen. So, this makes sense. And then we did some statistics in order to see whether, what type of bile acids, and there's a bunch of different types of bile acids that the liver makes, and these are the abbreviations. I come to 2 different species, types of bile acids in a minute, would correlate with a certain type of microbiota composition. Okay, the hypothesis would be that the secretion of a certain bile acid would drive a certain type of microbiome. Okay, could be, but how to test this? So the way we did this was now again to take neonates and compare them with adult mice, or to expose them to these bile acids and then look what the microbiome does. And so if we do this, then we this is the adult, I think. This is the adult, and this is the this is the adult and this is the neonate. So this is the untreated neonate. This is the untreated adult. So the microbiome, if you wish, moves from here to here. So it's a kind of a certain type of description of the composition. Now, if we give, for example, taurocolic acid, which is one of these bile acids, we get the microbiome in neonates closer to the one of adults, right? And the same is true if we give beta-tauromurocolic acid, and this is again one of these very dominant ones in neonates. We get this neonatal microbiome more in the direction, which is still neonatal. The mice are small, but more in the direction of an adult microbiome. Okay. And we also see that the richness increases, which is a typical sign of adult microbiota, and the similarity decreases, so it's somehow different. And since we know that one major difference between the neonatal and the adult microbiome is the presence of lactobacilli in the adult, which are normally very low in neonates, and the absence of something like Escherichia, for example, gamma proteobacteria, which are high in neonates but low in adults. And beta-tauromurocolic acid does exactly this. So it pushes lactobacilli numbers, so it makes it more similar to the adult, and it decreases Escherichia coli, again, more similar to the adult. And this is probably the way it works. So it kind of is able to push the microbiome in the direction of a more adult-like microbiota. There's no formal proof that this is indeed, but it's, what it's intended to do, but it does this. So, you could, again, this could be an example that the host indeed shapes the microbiome. But it's more complicated. And it's just an example just to show you how complicated this becomes, because the microbiota also influences the bile acids. There's one very famous type of modification, and this we saw in a model that we don't have to bother here, but it's a neonatal Giardia lumbria infection model. Giardia is this parasite that I showed you. We know Giardia because when we go to India and come back with diarrhea, we have a good chance this is Giardia. It's a very nasty diarrhea. It's lasting and difficult to treat, but it will, at some point, it will heal. It's interesting, in endemic areas in Africa and Asia, children in some areas have high incidence of Giardia. And here it's not diarrhea, the disease, but rather a metabolic disease. So they're smaller, these kids, and have less weight. So it's a metabolic phenotype. Now, this is why we did the mouse model, and we can replicate this. But regardless, when we infect with Giardia in these mice, we see a difference in the microbiome, and in particular of certain taxa like Coriobateria or Enteroptus, Coriobakteraceae or Enteroptus, which is the genus, that have this enzyme, which is a bile salt hydrolase. And this, what it does is it removes the amino acid from the bile salt, so it deconjugates it. And in particular in mice, and similar to neonatal humans, interestingly, and not adults like us, we have glycine conjugated bile acids, but human neonates and mice use taurine mostly as an amino acid for this conjugation. And what was interesting then, in these Giardia-infected mice that had this microbiome difference, indeed we saw a global decrease in taurine conjugated bile acids and an increase of the deconjugated bile acids. Okay, so what I basically want to say is that bile acids may be able to drive the microbiome, and this may be intentional and a mechanism to shape it in a certain way. But there is also the possibility that kind of the microbes kind of fight back and then change, the bile acid profile. And this again, may then have certain consequences. And infection here as an example to actually induce this. And now maybe come to the third question, which is, so what is the effect of the microbiome? And this is a big field, if you wish. Now with these microbiome researchers, maybe 50% of them are immunologists. Try to figure, what effect does the microbiome have on the immune system? Obviously an interesting question. And we've also been working on this. And I just want to remind you some simple things that it does. And this is, for example, colonization resistance, something very simple. So a microbiome that is less diverse has open niches, ecological niches. And so we can infect neonates very easily with pathogens, which is more difficult in adults. So if we take, and I give you two examples, one is pathogenic E. coli, and the other one is Salmonella. So with EPEC, for example, we can go into mice and colonize them right away without any treatment or anything. They're colonized nicely. And it's interesting to see that the more diverse the microbiome becomes, these mice at some point expel EPEC. So EPEC is lost. So this is a competitive colonization behavior.
And with Salmonella, we do the same thing. And here, the interesting thing, I mean, the same thing happens. This is a little model, so we cannot study long term. What we can do, we see that 100 bacteria, Salmonella would be enough to colonize 100% of these animals, completely, and they reach similar numbers after only 2 days in the colon. So very different to the adult model. Wolf knows this better than I do. But I mean, if you would do the same thing, you need, I don't know, 10,000 more bacteria, and then you get 50% of the mice being colonized, I guess, maybe. So very efficient. And this makes it possible that some models, you can do certain type of infection models in neonates. And this is, for example, the EPEC model, where we can see these typical hallmarks of EPEC disease, which are these attaching and effacement lesions on the epithelium with actin changes, and this is an electron microscopy. And now we're doing a lot of studies with these EPEC on EPEC, because that's understudied, because so far there wasn't any in vivo model for this. Just to remind you, the microbiome here impairs colonization in the adult. So if we don't have it, we can colonize it. Adult mice that have a microbiome would, just not be colonized by EPEC, but it's not sufficient for these little beautiful microcolonies, because germ-free mice or antibiotic-treated mice, even though we can colonize them, would not form these microcolonies. So there's a different reason for this, and we still trying to figure this out. Okay. Colonization resistance has a bad side. It actually has a good side, and Emma talked about this before. Probiotics. And I mean, if you go to the supermarket, you can buy probiotics. It's basically like yogurt, right? It's very sweet, a bit more expensive than yogurt. And it's basically, as adults, it's pretty much useless. And the point here is that if you take probiotics that are defined as viable bacteria that have a beneficial effect, they would just pass through your gastrointestinal tract, and they would have no chance whatsoever to colonize you on any kind of prolonged period, because there are already microbes that do much better than these probiotics. But the situation, and I showed you, gave you examples, is different in kids. And here we studied preterm babies, even preterm neonates. So we had the chance to have preterm neonates that did not receive any probiotics, or 2 different types of probiotic cocktails. One with kind of 2 bacteria, the other one with 4. And our question was, how efficient is colonization? And, it's not super efficient. It's not long-term, what we can say. But yes, we see kids we see in particular some strains more than others can colonize more than 2, 3, 4 weeks in these kids. So we can kind of, if you wish, in a sort of a permanent manner, colonize neonates, which we could never do with adults. What's interesting here is they do outcompete. Now, these probiotics protect from NEC, and Emma again mentioned NEC before, necrotizing enterocolitis, the inflammatory disease. And also in our study, that's been shown by others. We could also see this, right? The control group had some NEC cases, none of the treated group had, or I think there was 1 case. And certain type of bacteria seem to drive NEC disease. And in the treated kids, these, NEC driving, mostly gram-negative Enterobacteriaceae are reduced in their abundance in the gut after treatment. So this is probably the way they work. It may not be the only way they work. But this means there is maybe a good side of not having a competent microbiota, because you can still, modify it, manipulate it. I think kids are probably the best example if you want to manipulate the microbiome, which probably Emma wants to do. So maybe she's on the right track for this. And I have you a last example. I'm a bit over time. Can I say? Is it okay? Okay. And this is more on the adaptive immune system. And it started when we did immunostaining in the intestine of mice of different age, and we stained for T cell alpha beta T cells, so classical T cells. And what we see is very little T cells in neonates in the intestine, with the exception of Pias patches, where we can see these T cells being homing to. Only in adult mice we see then T cells in the lamina propria. It's not unexpected, but maybe the extent is still striking. So there's really nothing. If you look what we see in the neonate, it's mostly CD4 T cells and then only later CD8s. And what's interesting, this is nothing to do with the microbiome. Regardless of what I just told you, the colonization until this point has nothing to do with the microbiome. Whether we look into germ-free mice or colonized mice, it's just the same. In the adult, there is a difference, and this has been published many times. But we found that interesting, and then we looked more into these cells that colonize the neonatal intestine and wondered, when do they get actually activated? And when you do this, then we notice that actually this takes a long while. So in these Pias patches, they, from the transcription profile, are similar to thymocytes, so in the thymus, so very immature. Whereas in the lymph node, already at day 6, they group together with adult lymph node T cells, which are really different. And if you look into CD44 expression, which is more like a mature T cell, in this case, they stay low in there. The fraction of CD44 highs is relatively low and rises only in the adult together with germinal center B cells. So it seems as if T cells home to the gut, but then there is a break on them. So they do not mature. And until, after day 28, which is quite late. And I showed you before that the microbiome is there. There's antigen. There must be innate stimulation. Why is this? So we figured in a previous study, we figured that, there's a little bit of something of antibodies from the mother that helps to have the number down. And also like Tregs in the neonate seem to participate, but they do not explain the phenotype. So we kept doing. So we looked into this, and since we knew that it's not the T cells itself, because when we take out T cells from neonates and adults and compare them functionally, they actually and put them in adult mice and look how fit they are. They have the very same features. So we thought about now what's upstream. So what is the environment of these T cells in the neonates? So we took OT1 or OT2, so CD4 or CD8s, CD8s or CD4 T cells into either neonates or adults, and then gave the mice over and checked maturation. What we see is that the OT1s are fine, but the OT2s show a difference. So neonatal OT2s are much less activated. And then we did an in vitro assay, again, total Pias patch antigen presenting cells plus OT2 T cells, and looked into the cytokine production. And this was kind of consistent because now we saw less IL-4, maybe a little bit less IL-13, but more IL-17 and interferon gamma, which is consistent with a maybe reduced type 2 immune response, but more 1 and 3, which again is consistent with the fact that it's a bit difficult to vaccinate neonates. And maybe that's interesting because that's a lot of translational efforts go in this direction to improve vaccine efficacy in neonates. It's a major problem in public health. And so we said, okay, that's interesting. Now let's look into this environmental factor, which is probably the antigen presenting cell. And so we sorted antigen presenting cells from the Pias patch of adult and neonate mice and compared them by single-cell RNA-seq. They fall into different clusters like CDC2s and CDC1s, and they come in a quiescent and an activated stage. We have our gamma T APCs, which is a bit particular, and then monocyte-derived antigen presenting cells, so lysomax. And when we so they're all present. If you look into the numbers by FACS, we see that in particular the CDC2s are actually underrepresented in neonates, whereas CDC1s and our gamma T APCs are actually overrepresented in neonates. This makes the CDC2s interesting. So when we do differential gene expression, we find certain genes, and if we do Joe terms, we see something maybe a bit expected, that in a neonate, we have mostly cell cycle and differentiation genes, and in the adult, antigen presentation and processing. So it seems to be a maturation difference between the neonatal DC2s and the adult DC2s. So this asks for looking into this by pseudo time. And so we did. So, and then you see indeed that in the neonate, which is the blue, you have an overrepresentation of more like immature stages. And here to the right, we get to the most mature stages. We have an overrepresentation in the adult. We can also now look for markers and then define different stages. And then we can, again, by FACS, try to confirm this. And again, we see that in the adult, we have more the mature stages, and in the neonate, more the immature stages of these DC2s. And now we get back to the microbiome. There is a microbiome effect on this, because in germ-free mice, this is shifted, not stage 1, but stage 2. We have less in germ-frees and more in germ-frees, and stage 3 less in germ-frees. So there seems to be an effect driving maturation, at least from step M2 to M3. This is very nice. The R gamma T iPSCs are much less cells, so it's been more complicated, but we see a similar phenotype. So we have more in the neonate of the immature stage. And if we compare, and now we don't have the staging, but rather just the age, if you compare germ-free with conventional cell mice, we have less of these RA gamma T APCs in the neonate, rather more in the adult, which is interesting, and that's been published. But we have less in the neonate, and that's wasn't published. So how could we use this? If it's a maturation defect of DCs, maybe could we change this? Could we interfere with this in order to drive maturation and get better responses? And what we noticed was a remarkable interferon response in these CDC2s in the differentially expressed genes. And so we thought, what about if we induce interferon and interferon, expose them to interferon, could we drive DC2 maturation and then the response? And so we did. So we took mice, treated them, in this case with R848, which is an interferon inducer. Neonate mice have lots of PDCs, so they make a lot of interferon in response to R848. And this is what you see here. So there's lots of, they react enormously to this with many, many cytokines, among them type 1 interferons, actually more than adults. We also see now the ISG signature, so it seems to work. And if you look into maturation, it again seems to work. We see in these treated mice, the dark blue, many more of the mature stages as compared to the immature stages. And this is, of course, important, we also see that the OT2 response of those mice that are treated now is higher, right? Which was, before I showed you, it's reduced in neonates as compared to adults. When we treat them with R848, this response goes up, as does the gamma production, gamma positive CD4 T cells. So we think that, with interferon induction, maybe we can drive DC maturation. And maybe this is the problem of these neonates that impairs the immune response to vaccination. And with this, I would like to finish and just thank my group that actually did the work and the funding agencies. And we've got help from many collaborators. And with this, and thank you for your attention.